So let's talk about the heart. So the heart has specific layers. Now there are typically three layers: the endocardium, the myocardium, and the pericardium. Now you also might have seen the term epicardium. This is because the pericardium actually has two layers: the visceral layer and the parietal layer of the pericardium. Now the visceral layer also is commonly called epicardium. And of course, these two layers, the visceral layer of the pericardium and the parietal layer of the pericardium, are nothing more than just two layers of mesothelial cells that produce specifically a fluid, viscous fluid that prevents friction. Now, it's also true that the pericardium contains adipose tissue. Again, the purpose is the same, to reduce the amount of friction from the heart to the other organs, or also to heart structures such as the rib cage. Now, or the lungs and so on. So first off, let's start seeing about the different layers. The most important layer, of course, of the heart is the myocardium, the one that exerts the function of contraction, rhythmic contraction, and of course its purpose is to again to force blood from the system towards the pulmonary tissue and vice versa. So we already discussed the contents of the myocardium in the muscle video, but just a short recap of what was being said. So the cardiomyocytes are again cells that are typically single nucleated, maximally 2 nuclei per cell maximal, and this cell is characterized by the presence of desmosomes and gap junctions along the intercalated disc. Now the intercalated disc is a structure that connects one cardiomyocyte to the other. It's important to understand that within this, or throughout the whole length of the intercalated disc, we're going to find both desmosomes and gap junctions. The function of the desmosome is already known, and of course it is to achieve intercellular adhesion, so it has to maintain the two different cells attached and close to each other. And of course the function of the gap junction is to transfer molecules. Now in case you don't remember this, the purpose of the gap junction is to transfer molecules, but the limitation that these molecules must be of small size. It can transfer big proteins, can transfer big molecules. For example, this is an ideal scenario in which we can actually outline and underline the importance of gap junctions, because they can actually transfer specifically ions. And of course, this in the chemical world, the ions, in the biochemical world, the ions are actually also part of the signaling of cell signaling and the transduction of the signals and the transduction of the cardiac pulse. So it is important that we have the electrical propagation of the nerve of the signaling from one cardiac muscle cell to the other cardiac muscle cell through the gap junction. In other words, gap junction is a structure that actually ensures the contractile and rhythmic contraction of the whole ventricles and the whole atria and the whole heart as a unit. If we have any sort of obstruction or any sort of malfunction or a group of cardiac muscle cells, we actually can end up having a cardiac pathology, also called arrhythmias. This is, for example, why after an infarction, which is the formation of a clot and the death of a specific area of the heart, we have this. It's very common to have arrhythmias after an infarction. This is because a group of cardiomyocyte cells in a specific location are dead and no longer can actually propagate the electrical signal coming from one part of the cell and through the gap junction to another cardiac muscle cell. So let's focus just a bit more on the conducting system. What does it actually means? So the conducting system is actually, this is a topic mostly for physiology, but a quick sum up is going to be just what we're going to do right now. First up, we have the pacemaker, the sinoatrial node. This is the one that actually initiates the electrical impulse, and again, of course, as we can see in the picture, let's say propagates it throughout the atria. Now, the second node that we should actually take mention of and pay attention to is the atrioventricular node. This node, of course, can function as a secondary pacemaker in case that the first one is not functioning properly, or there is not proper rhythmicity and let's say proper synchronicity between these two. This actually will initiate the electrical conduction signal itself. Now, after the atrioventricular node propagates and it creates this signal, this electrical signal, it's going to pass throughout the whole atrioventricular bundle, or also called bundle of His. In the end, of course, all of these nodes, all of these bundles are going to end up with the smallest unit of the conducting system, the Purkinje fibers. Again, not to be confused with the Purkinje cells of the nervous system. So the Purkinje fibers, of course, all the conductive system, and specifically the Purkinje fibers, are also themselves cardiac muscle cells. They're specialized cardiac muscle cells, specialized in conduction, but they're also don't forget they're also cardiac muscle cells nonetheless. There are major differences, some difference between the Purkinje fibers and the typical cardiac muscle cell, but we'll actually underline them right now. Of course, one common finding is that the bundle of the Purkinje fibers contain high amount of glycogen. So this actually is a very, very interesting and very important point. Why? Well, because presence of high amount of saccharides typically make a specific structure stained through other stainings, for example, special stains, for example, the PAS method, the PAS method. We're going to see in a bit the differences between the microscopic differences between the Purkinje fibers and the cardiac muscle cells. In typical hematoxylin and erogen, H&E staining, it's not very easy to visualize the differences, and that's why you actually utilize the PAS staining to specifically and directly identify which of these cells are the Purkinje fibers and which is the normal cardiac muscle cells. So let's actually see the pictures of the slides of the heart. So first up we have the endocardium, the innermost layer that is actually in contact with the blood. Now the endocardium is very, very similar, in fact, to the tunica intima, the same as we saw in the vessels. So first up, if we zoom in, we're gonna find again the endothelium, the endothelial lining. Now again, the functions are exactly the same. The simple squamous epithelium, the endothelium are exactly the same. The endothermogenic activities, and so on and so on and so on. So in this case, we're going to find the endothelium, the innermost layer of the heart, and we can find the very, very nice simple squamous epithelium. Again, the functions are the same of the tunica intima, the endothelium of the vessels, and of course, exactly beneath always the simple squamous epithelium and all sorts of epithelium, we can find the lamina propria, the subepithelial loose connective tissue. Now here there is one difference than the others, than the typical other loose connective tissue. Here we also find sometimes interspersed smooth muscle cells. Very few, not common, but also can be found. It's a normal finding. And of course, the biggest and thickest layer of the heart is the myocardium, the one that exerts the contractile function of the heart. This is the most important layer functionally of the heart. We've already discussed the morphology of the cardiac muscle cell along with the function and the morphology in the video of the muscle. Feel free to go back to refresh your memory on this. And lastly, we have the epicardium. Now the epicardium is the outermost layer, and again it's lined with the two layers. Again, we'll discuss the epicardium is the visceral layer of the pericardium, and of course the epicardium is the visceral layer, and of course they're also the parietal layer. So there are these two, let's say, layers. One and second, both of them are mesothelium. Again, the term is different, mesothelium, because we have actually specific unique functions. And here the unique function of these mesothelial cells is just to produce this viscous fluid that actually reduces the friction between the heart and other organs or heart tissues. So it's not uncommon to find adipose tissue between the layers of the outer layers of the heart, and this is of course due to this is a normal physiological presence, and this doesn't denote pathology, but of course excessive amount of adipose tissue in the heart exerts excessive mechanical work for the heart workload, and as a consequence, generally speaking, whenever they are forced to do something extra, either extra work or some parafunctional habit or something that is not out of like the nature of the cell, then they have two typical reactions. Cells that can divide, are able to divide, they typically undergo hyperplasia, and cells that typically tend not to divide and do not have this mitotic potential, they typically tend to hypertrophy. And because of the heart to undergo hypertrophy, and because of the cardiac muscle cells do not have the potential to multiply and to differentiate, of course, the very typical reaction of the heart when exposed and when the workload of the heart is excessive is to hypertrophy. So in this case, whenever we have this excessive thickening of the heart, we typically have conditions such as cardiomyopathy, restrictive cardiomyopathy, where the thickness of the myocardium is excessive, and this is because of the increased workload, and as a consequence there is smaller space for the heart to receive blood and to pump out, resulting into many other consequent pathologies. Now, as I said before, we have this Purkinje fibers. The Purkinje fibers, of course, they're part of the conducting system of the heart. And we already said that we have their PS positive. So here we have the staining of the PAS method, and we can actually visualize easily the difference between the normal cardiac muscle cell and the Purkinje fiber. Of course, we can find the very similar appearance in the H and E, but in the PS method, we can actually visualize the vivid pink colors and the positivity in the PAS methods throughout the whole layer of the Purkinje fibers. Now, exactly beneath, we can find the typical cardiac muscle cells, which of course do not contain a high amount of glycogen, and as a consequence are not positive to PAS staining method, of course, in comparison to the Purkinje fibers. Thank you very much.